Good afternoon. My name is Dr. John Thomas for the University of Texas Rio Grande Valley Department of Human Genetics within the School of Medicine. This will be the short course session for the Center for Genomic Research. This is short course session number 4, Neurotropism, the impact of molecular interactions on the nervous system. So in the previous 3 short course sessions, we've discussed the fundamentals of immunology. We've discussed the immune response within the context of viral infections. And we did a brief discussion on viral genetics and how certain viral diseases can elicit specific disease types within individuals that bear certain autosomal recessive or autosomal dominant gene deletions. We talked about inborn errors of immunity, specifically for viruses that tend to have a latent replication aspect as part of the replication cycle. So we specifically talked about herpes virus, varicella zoster virus, and then other viruses such as Epstein-Barr virus. So today, to wrap up the short course session for the 2024-2025 academic year for the Center for Genomic Research grant, we're going to discuss neurotropism with a specific focus on certain what is neurotropism, how do we elicit a disease state within the brain and the central nervous system, and we'll look at some viral diseases, parasitic diseases that can cause that have displayed neurotropism. And we'll also look at some prion-induced pathologies as well, and really focusing in on diseases that are going to be prominently infecting the brain and the central nervous system and how they establish this disease state within a healthy individual. So let's briefly start with some terms. So what is neurotropism? So neurotropism is essentially a fancy word that we use to describe a tissue within the body range, a tissue range within the body for a given pathogen that has a preference for these tissues, specifically tissues that are involved with the neural lineage and organ systems of the central nervous system. So the technical term or technical definition, if you will, is neurotropism is the ability of a pathogen to invade and survive in the central nervous system. So if we look at the range of pathogens that have the ability to interact with, penetrate, and infect the central nervous system and the brain as a component of the central nervous system, there are certain viruses that can do this, dengue virus, Zika virus. These are not the only exclusive pathogens, but these are just some examples. There are certain parasites, Toxoplasma gondii, Entamoeba histolytica. These are certainly pathogens that you see sometimes from people that swim in bodies of water that are very still. Sadly, this is a disease that we see and a pathology that we've seen pop up, particularly during the summer months where it gets a little hotter. People are more prone to going outside and swimming in natural bodies of water. So certain parasitic diseases can have neurotropism and can cause severe debilitations to individuals that are infected. We have certain types of bacteria, strep pneumonia, Listeria monocytogenes, and even certain forms of cancer that can all display a strong preference for establishing their infection or their disease state within the central nervous system. There are going to be differences, of course, because these are different pathogens in terms of how they establish their infection within the central nervous system. But the basic mechanism for invading and being able to establish a foothold, if you will, in the CNS is very similar. So how does a pathogen first cause a disease state within the central nervous system? So how do we get this? Well, one of the main functions that we have, anatomical barriers that we have that functions to protect our central nervous system from pathogenic infections, from disease states, is called the blood-brain barrier. And this is actually a physical barrier that, for all intents and purposes, encompasses the brain and provides a physical wall, if you will, that precludes the brain from being exposed to everything else that's in the body. So if you think of this, it's almost like a force field that goes around the brain, and this force field is composed of not only very tightly joined epithelial cells, layers of epithelial cells that are very tightly joined and held together, but we've also got a flow of fluid and some other formations within there that all help to very tightly encapsulate the brain structure and keep the brain from being exposed to pathogens. And this also allows for there to be a homeostasis, right? The body wants to be in equilibrium as much as it can. So this also allows the CNS to enact some kind of non-specific homeostatic effect which allows for proper function of the neurons and protects the brain from toxins and pathogens. You can imagine if every time you got a cold, the cold and the immune system responding to clear the cold infection was able to get into your brain, you then would be in a situation where you might have cells of your own nervous system responding against your brain tissue, trying to kill brain cells because the immune system thinks that these may be cells that are infected with pathogens. Remember, we talked about in the introduction to the nervous system in short course session number 1 that we have subtypes of cells within the body that comprise the immune system, primarily B cells and T cells, specifically T cells, whose job it is to go in and identify and eliminate virally infected cells and cells that are infected with intracellular pathogens. So if you got a cold infection, for example, you got a cold virus infection and the virus was able to infect cells of your brain, you would then be in a position where you might have your own immune system going in and killing off your brain cells. Obviously, this is not something we would desire for a good outcome. So the purpose of the blood-brain barrier, or the BBB, is to act as this barrier that's not going to prevent pathogens from getting across it. In a normal, healthy person, this barrier is going to be intact. It's very robust, and it's not going to be able to be penetrated by most pathogens. So if we look at if we look at the images up here, we've got a CT with contrast and without contrast. If you look at the outermost layer right here that's identified with this bifurcated line that's encapsulated by these 2 white layers, this is the blood-brain barrier. And these are very tight epithelial cells held together with very tight junctions. They're going to almost form a protective layer around the brain. And the job, again, of the blood-brain barrier is to prevent pathogens from gaining access to the brain and then to the central nervous system. So if a pathogen wants to establish infection within the central nervous system, the first task it has to accomplish is it's got to be able to circumvent the blood-brain barrier. If we look at some additional images here of some of the structural features of the blood-brain barrier, what we see are these ECs. ECs is going to stand for epithelial cells. And within these epithelial cells, within each individual epithelial cell, they're going to be connected by these very tight junctions, abbreviated by TJ. These tight junctions connecting the epithelial cells are going to form a very strong contiguous barrier of cells that are going to go around and encompass the entire blood-brain barrier. If we look at some of the additional cell types that make up the blood-brain barrier, we see PC and AC. We see periocytes, we see C, which are the astrocytes, these AEs right here. So we have some different cell types in here, including astrocyte end feet. We've got basement membranes, which are indicated with BMs. And we've got a system of cell types that are in here that all are comprising the blood-brain barrier that are going to work to give us this intact, contiguous structure that's going to protect the brain and the central nervous system from infection. So in a normal, healthy individual, this intact blood-brain barrier is going to keep pathogens away. Now, as you probably remember from the short course session 2, where we discussed viral genetics and viral replication, we have a wide range of viruses that are using different types of genomic material, either RNA or DNA, to be able to get in and replicate within a specific cell type. And just to kind of refresh everybody again on the life cycle of viruses, the virus has got to come in. Some of these viruses have got very complex genomes that have got to be processed by both host cell and virally carried proteases. The virus has got to be able to attach to a cell, to a permissive host cell. It has to be internalized, which has got to penetrate and get across the plasma membrane. And the genome has to encode so it can establish infection. So this is just a very 10,000-foot view, kind of going over again what we talked about in short course session 2 about virus replication and viral life cycle. So you've got a virus coming in that wants to bind to a host cell. It's got to bind to that receptor on the host cell to be able to gain entry, to penetrate, and then be able to uncoat. So again, the blood-brain barrier's goal is to prevent this entire process from even occurring, because it's going to prevent the virus from even gaining access to the cells that are underneath the blood-brain barrier. So the neuronal cell tissue, the cells that comprise your brain, the blood-brain barrier's job is to prevent these viruses from getting access to it. Nevertheless, we do see infections occur that are able to cross the blood-brain barrier. One of the viruses that's recently been identified as being a virus that we refer to as being neurotropic. This means the virus has the ability to infect cells of the neuronal cells from the neuronal lineage, specifically cells that are underneath that blood-brain barrier. One of these viruses is dengue virus. Dengue, up until about the early 2000s, had been considered to be non-neurotropic in humans, but several studies looking at case reports that came out of Japan and Korea in the early 2000s have described the presence of dengue virus in the CSF and brain tissue of certain individuals coming in. And a recent study in 2011 looked at the potential for dengue virus to cause encephalitis and behavioral changes through attacking and infecting the cells, the neuronal cells that are underneath the blood-brain barrier in mice. So this image that we see here is an MRI that was done on an individual that presented to a hospital in Seoul, South Korea in 2008 that had a dengue virus infection. Normally, this is a normal adult MRI for a brain. And here again, we see our blood-brain barrier. We see a very nice looking brain overall. We can see the gyruses, we can see the folds and the succinates. We can see some basic structures of the brain. This looks like a good brain. When we look over here, this is an MRI that was taken from a 44-year-old man who was infected with dengue virus. And you could already see when you look at differences in the blood-brain barrier, we're seeing a dramatic reduction in the overall size and thickness of the blood-brain barrier. We're seeing a bit of a breakdown in some of the underlying layers of the blood-brain barrier. And if we look a little further here, it appears that we're starting to see a little bit of pullback and reduction in the overall size of the blood-brain barrier. So this is from an individual that was shown to be infected with dengue virus. Virus was actually recovered from his CSF. This individual did later die from dengue virus, and so an autopsy was able to go in and sample and stain some of the brain tissue and positively identify dengue serotype type 2 as being the cause of the infection. So when a similar study to replicate the human dengue virus infection was repeated in mice, what the researchers found was that dengue virus did appear, and they used dengue type 3, dengue virus type 3 serotype to be able to do this infection in mice. But what they found was that virus was able to penetrate the blood-brain barrier, and they think that this occurred through a process where leukocytes mediated the infection by going through a process called rolling. So there's a mechanism that describes how cells move in the body, specifically certain cell types, and this has been very heavily studied in the immune system. So leukocytes in particular go through a process that we call rolling and tethering, where they essentially, if you could imagine the cell as a sphere moving through space, it has its little projections on the outside of it. And as the leukocyte rolls through space, it's able to grab onto something, adhere to it, and then use that to kind of swing itself with some momentum along its journey. We call this process of grabbing and holding and then swinging, rolling and tethering. The tethering is where the leukocyte grabs onto a cell projection. The rolling is the mechanism that we use to describe the transfer of momentum that allows the leukocyte to kind of continue moving through three-dimensional space. So what these researchers found was that when they looked at dengue virus type 3 infections in mice in the brains, they found that leukocyte rolling and adhesion seemed to preclude the infection with dengue virus in these mice. So in the control animals, they were able to block or abrogate in large amounts the ability of leukocytes to roll and tether. But when they looked in groups where they did not block this leukocyte rolling and tethering, they found significant increases in dengue virus, and they found dengue virus in the brain. When they looked to see what were some of the effects of dengue virus replication in the brains of these mice, what they found was that they saw severe behavioral changes. So they saw losses in muscle tone and strength, as indicated here. These are the control groups right here. This is an infected group right here in the triangle. As you can see up here in the legend, the square is a sham, the triangle is infected, and we see that both losses in motor behavior and muscle tone and strength were highly elicited in the group that was infected with dengue virus. However, the sham group up here in the top maintained muscle tone and motor behavior. So this study showed that, at least in mice that were infected with dengue virus, did appear to elicit and exhibit some neurotropic disease factors as a result of this infection. And as you can see over here from the control, we see that in terms of survival, the control animals, we had 100% survival, but animals that were infected with dengue 3, all of them, death started at about 7 days post-infection, and by 8 days post-infection, all the animals were dead. This was an N of 20 in both of these experiments here. So if we look at some of the clinical manifestations of dengue virus, which belongs to the flavivirus infection, we see a very similar pattern. We see rashes, we see petechial rashes, particularly on the face. We see rashes in the extremities, and again, this is going to be very similar for dengue, for Zika. We most commonly, though, do see these rashes forming in dengue viruses. But as I said earlier, we know that there are other members of the flavivirus family that do display some incidences of neurotropism. So dengue virus's ability to infect humans, to infect the brain and central nervous system of humans, as well as in mice, has, I think, received in some recent years some good evidence. But there's another virus that's out there that we know is very well established as a neurotropic agent and can lead to very severe pathologies if it's able to get into the brain by penetrating the blood-brain barrier and replicate uncontrollably. And that virus would be Zika virus. We know that there are other viruses besides the flaviviruses that can cause neurotropism. The alpha viruses, which is characterized by certain members such as Semliki forest virus, Venezuelan Western equine encephalitis virus, chikungunya virus. These are all viruses that are known to infect humans. They also infect mosquitoes, so they alternate in a zoonotic cycle between a mosquito host and a human host. But the alpha viruses have been very intensely studied for their ability to cause neurotropism. And as these images are showing up here, these are brain cells that have been stained with alpha virus clones that are expressing GFP. So all the green fluorescence that you're seeing here is viral replication, and these are different slices from the hippocampus and staining of individual neurons showing that alpha viruses do in fact infect the brain tissue as well. So viral infection within the brain is not an uncommon feature, unfortunately. And again, all of these viruses are going to be able to establish infection by penetrating and getting across that blood-brain barrier. Yeah, just kind of confirming that we see this in neurotropism in many different viruses, not just limited to dengue or Zika virus. Of course, Zika is probably one of the most famous because of the Zika outbreak that we had in 2015, where we saw a dramatic increase in cases of microcephaly in Brazil. Brazil had less than 250 cases in 2014. In 2015, they had over 3,500. And we still to this day do not know exactly what was the cause of this dramatic spike in microcephaly amongst the population in Brazil. During the outbreak in Brazil in 2015, there was also a concurrent Zika outbreak happening in Colombia and in Argentina, but those countries did not see, for whatever reason, a dramatic increase in the cases of microcephaly. But again, if the virus is able to get across the blood-brain barrier and penetrate, then it's going to replicate inside of neuronal tissue. And as we saw with the microcephaly outbreak in 2015, virus replication inside of the brain, the CNS, is going to be able to largely proceed unchecked because the immune system generally does not have the ability to access the brain to go in and create a sterilizing immune response. So we still to this day, 10 plus years after the Zika outbreak in 2015 in Brazil, have not really identified a definitive link. But there is a consensus that Zika virus does induce severe pathology in the brain. We have a lot of clinical data and animal data that shows this. And so we're really now just trying to see if we can identify what the specific link is between the two. But again, when we have virus replication unchecked inside of the brain, you can see here, this is a baby, an image of a baby with microcephaly. This is a baby with a normal head. We're seeing about a 40% reduction in the size of the head because we're seeing a dramatic reduction in the overall size and organization of the brain. This is an MRI from a normal child. This is an MRI from a child same age, both about 6 years of age. This one has microcephaly, and you can see the dramatic differences between the two. You can see complete loss of definitive structures, right? This is the hippocampal area right here, the hippocampus region. That region is largely absent here. You can see how the brain has largely remodeled itself. The pons efferens, which we would normally see a little bit closer up top to the skull, we see back here is stretching all the way back. we see a dramatic pullback in the brain from the blood-brain barrier around here, and an overall reduction in the size of the brain. So we know that viruses have the ability to cause neurotropism by getting across the blood-brain barrier. So it's definitively established that we know viruses can do this. Now what we're really trying to figure out is how viruses are doing this. One way that we've been trying to identify this for Zika virus is using a possum model of infection, where we've infected the possums with Zika virus directly into their brain, and then we look for the presence of virus replication, and then we study the animals long-term to see what kind of debilitation or effects on the animal's body or brain we're going to see following infection. All the red dots in these images, these are different slices of neuronal tissue taken from the possums, showing that in each of these, as indicated by the red dots, this is indicative of Zika virus replication. So we do see Zika virus replicating in the brains of possums. This is the control animal over here, but all the other experimental animals that were infected in panels A through D are all showing the presence of Zika virus. So we know that we can infect the possums intracranially, and when we let these animals proceed along for 60 to 90 days post-infection, what we begin to see is a bit of a spongiform morphology in the brains of these animals as they are allowed to persist with a Zika virus infection. So these are control animals. All these animals were at 90 days of age. These are control animals over here on the left, and you can see, looking at the brain tissue, it looks very nice. For the most part, this appears to be a single contiguous layer, not a lot of gaps in there. There are some over here. This is the same tissue at a little bit higher resolution. This is at 60x, this is at 100x. But again, for the most part, you're seeing tissue that you would expect it to appear somewhat normally. When we look at the infected animals, however, we already start to see at lower magnifications a spongiform morphology, where we're seeing holes in the brain tissue. Remember, this is what the brain tissue should look like. This is what the brain looks like after it's been infected with Zika virus for 90 days. And if we zoom in a little bit further and stain for the presence of virus, we find in the same tissue at 60x at 100x magnification, we begin to see presence of these red dots, which indicates Zika virus replication. So we've been able to establish in our possum models that not only can Zika virus replicate and persist in the brain long-term, particularly in the absence of a sterilizing immune response, but the virus can also elicit profound changes to the brain tissue morphology, as indicated by this spongiform appearance that we see in this animal. And this animal was representative of about 10 different animals that comprise this group. In addition to causing degradation and breakdown of the brain tissue, we also see an increase in apoptosis as a result of viral infection within the brain tissue. And just as we spoke earlier about how negative it would be for the host to have an immune response directed against virally infected cells within your brain, having massive numbers of cell death, also referred to commonly as apoptosis, occurring in the brain at the same time, is also something that's not going to be very beneficial for the host. So again, if we look here, these are in panel C and D. This is a control animal, where this is a normal slice of the brain at 60x. This is a slice of the brain at 100x. And the red in these images are going to be indicative of apoptosis. So we do see some level of apoptosis, which would not be unexpected as a normal consequence of infection. However, when we look at panels A and B, these are animals that were persistently infected with Zika virus over 90 days. And when we look for apoptosis, we see, compared to the controls, massive amounts of apoptotic cells, many more, significantly more than you see in the control animals over here. So again, this just kind of further confirms that once you have viral penetration of the blood-brain barrier, you're going to get unchecked viral replication. Apoptosis is going to be a consequence of infection, and this is all going to lead to the breakdown and degradation of the neuronal tissue in the brain, which logically will lead to a state to where the animal, or if this was a human being infected with Zika in the brain, is going to display severe limitations and reductions in brain function and capacity. And we're not just, again, talking about the ability to play on the piano or recite poetry. We're talking about basic functions such as the ability to walk, to talk, to eat, even to breathe in some instances. So severe complications can result as a consequence of viral infection in the brain. And this all comes back to being able to penetrate and get through that blood-brain barrier. So if we look at microbial and parasitic neurotropism as well, we're talking about bacteria that can also get across the blood-brain barrier. We're going to see very similar outcomes in terms of tissue destruction and apoptosis and kind of a general lack of the ability of the body to clear these infections. There are some treatments and prophylactics that are available to you for use, and these are described here below. In many examples, when you have, particularly when you have encephalitis or a viral infection in the brain, it really is going to be a matter of time. And this kind of comes back to this conundrum in virology where when someone's infected with a viral disease, half the battle becomes, can we keep this individual alive long enough to give their immune system time to be able to respond to the pathogen? Because there is a severe lack of drugs that are able to directly go out and target viruses and remove them and destroy them from the body. So a lot of times the viral infection is just going to have to resolve itself over time as the infection runs its course. And then it becomes a case of, can we provide enough palliative care to the patient to keep them alive long enough to give themselves time to clear the infection. For bacterial and parasitic infections of the brain, some of these can be treated medically, but only with drugs that will target the infection. So they can't do anything to target the infected cell, but they can target the bacterium itself. So for viral infections, and some of these have probably heard of before, we have acyclovir, ganciclovir, forscarnet. Some of these drugs are used or in the prescription of treatment for herpes virus infections. Herpes virus is a virus that shows a specific propensity to infect neuronal cells, as well as rabies does. And so there are some drugs out there, like the acyclovirs, like the virs, acyclovir and ganciclovir, that can target the infection and try to help reduce the viral load. For parasitic infections, we're primarily talking about infections to go after certain types of neuronal parasites, Toxoplasma gondii, Entamoeba histolytica, pyrimethine can be given, plus sulfatidazine. These are also drugs that can be given over a course of about 7 to 10 days that will target the parasite. However, these drugs will only target a specific stage of the parasite's life cycle, and they can't get rid of encysted parasites in the tissues, so you've got to give multiple rounds of therapy. Oftentimes in parasitic infections, what we see is that when the parasite establishes the infection, what it does is it basically creates a zone within the body, if you will, where the parasite kind of makes itself immune to the host immune response by building a wall of dead cells and tissues around it to basically keep the immune system away from it. It forms this cyst. And this is a very common strategy that a lot of intracellular parasites use. Malaria does the same thing. And when you have these cysts formed, they encapsulate the parasite and make it very difficult to clear the infection. So oftentimes you're going to have to give multiple rounds of therapy to clear the parasite, because again, the drugs are only going to be specifically able to target a moment in time, if you will, as a part of the parasite's life cycle when it replicates. And so you've got to kind of continue to administer drugs over and over and over with the hope and understanding that you're going to be able to clear the infection slowly over time, because every time the parasite replicates, it's going to expose itself in that part of its replication cycle. You'll get some of it you'll miss, and you'll have to come back and do it again and again and again. And for the bacterial infections, it's really going to be any drug that can target the bacteria for elimination. And again, this is going to be something that you're going to have to do long-term, and you're going to have to administer palliative care. Unlike the viral infection, the bacterial infection is not going to resolve itself. So you're going to have to provide long-term antibacterial therapy and long-term palliative care. There are a new class of therapeutics that are out there that are in stage 1 and stage 2 clinical trials right now that are focusing on the ability of blocking the pathogen in regards to its ability to directly target and damage the blood-brain barrier, either by using some kind of a molecule that mimics the receptor that the pathogen would target, or to enhance the immune response through agglutination. So ultimately, if we kind of summarize this from a 10,000-foot view, the establishment of a disease state within the central nervous system comes down to the pathogen's ability to abrogate, to circumvent, to get around the blood-brain barrier and establish the infection. So long as the blood-brain barrier remains intact, the brain is going to remain safe. If, regardless of whether the pathogen is viral, bacterial, or parasitic, the BBB has got to be broken down in some capacity such that the brain is now exposed directly to the central nervous system. The blood-brain barrier can and will repair itself over time. This is why, again, if you're infected with a viral pathogen in the brain, most commonly the approach is give the person enough care to keep them alive so they can clear the infection on their own, and the BBB will repair itself over time. But as we see in many examples of infection, it often becomes a race against time. Can we keep the pathogen load in the host to an acceptable level such that the immune system can manage and clear it, while at the same time giving the host the space that they need to survive and persist long enough to allow the immune system to function and clear the pathogen? So for the second part of this, the short course session number 4, we're going to look at some specific diseases that affect the central nervous system, but they use kind of a unique mechanism. And so what we're specifically going to talk about now are prion diseases. So we're going to go through what are some of the different prion diseases, discuss how they affect brain functions, go over some basic components of prion biology, what we know about them so far, and lastly, we'll end up with some treatment options and some of the roadblocks or hassles that may come across with trying to treat prion diseases. So what are prions? In a very simple term, prions are infectious proteins. The fancy term is proteinaceous infectious agents, but prions are essentially infectious proteins that can be transmitted through the intake of this infectious protein. There are certain diseases that we refer to as transmissible spongiform encephalopathies. Again, if we look at the name, transmissible, this means that the prion disease can be transmitted from one individual, from one organism to another. And when we look at the pathology it causes spongiform encephalopathy. This is basically going to be that spongiform appearance that we saw in the possum's images about 3 or 4 slides back, where the brain looks like Swiss cheese. So we have transmissible spongiform encephalopathies, we have prion diseases that can be directly transmitted, and we have prion diseases that can be inherited. And within some of these inherited diseases, we're going to talk about Creutzfeldt-Jakob disease, new variant Creutzfeldt-Jakob disease. This is often referred to commonly as mad cow disease. We'll talk about some practices that led to the discovery of prion diseases. This is going to be ritualistic cannibalism from the Kruger tribe. And we'll talk about fatal familial insomnia as some of the main prion diseases that we see in humans. And then, of course, because animals are involved in this, particularly because some of these animals are either directly or indirectly in our food chain supply, we'll talk more about mad cow disease, scrapie, which is a form of spongiform encephalopathy that we see in sheep, and then chronic wasting disease, which is often seen in grazing ruminants. These are animals with hooves, such as deer, elk, and moose. And as you can see from the image here at the bottom, we will, of course, spend some time discussing cannibalism and how studying cannibals led to the discovery of prion diseases. So what is a prion disease? It is essentially, what is a prion? It is essentially a protein that is misfolded. As you probably remember from your molecular biology, protein shape determines function. Each protein in our body has a specific shape, and that specific shape gives the protein its function. If a protein loses its function, it's because it's lost its shape. So if a protein changes shape, it's going to change its function. In normal conditions, we have a protein in the body called the prion protein. It's a normal cellular protein that's involved in transporting ions within the body. It's very prominently expressed in the central nervous system. So prion proteins are expressed at high levels in the central nervous system. So maybe it's not a surprise that we see a lot of prion diseases associated with pathologies in the brain and the central nervous system. That's where the protein is primarily expressed. So when the protein is being expressed normally, we refer to it as PRP, when the protein is folded properly. However, something occurs with some mechanism, which we don't know why yet, which causes the prion protein, the PRP protein, to misfold. And when it misfolds, we give it the nomenclature of PRPSC. I think an easy way to remember this is PRP scrapie. The scrapie is one of the prion diseases that we see infecting sheep. So the misfolded prion protein is now turned into this mutant protein that we call PRPSC. Again, we don't know why the protein misfolds. But one of the unique things about prion proteins is that once they misfold and they take on this PRPSC, this misfolded configuration, they're now able to cause other normal copies of the prion protein to misfold as well. So in many instances, we can kind of think of this as an infectious protein disease. This protein is a normal protein with a specific shape, and then through some mechanism, it misfolds and loses that shape. And once it's lost that shape, it's able to basically pass that misfolding along to other prion proteins and cause them to misfold as well. Normally, a misfolded protein in under normal circumstances isn't a problem. There's a mechanism within the body, within cells, to break down proteins using the ubiquitosome and ubiquitination using the proteasome, and proteins can be broken down and chopped into their individual amino acid components and then recycled for other uses. However, the prion protein is very unique. When it misfolds, it becomes highly resistant to heat and digestion, so resistant that you can't break these proteins down in the body. You cannot even break these proteins down using common industrial processes such as autoclaving, which is used to sterilize surgical instruments. So an autoclave is essentially a machine that you can put metal objects into that uses very high temperature and very high pressure to essentially squeeze pathogens off of surgical instruments. Prion proteins are resistant to this process of autoclaving. So you can't break them down with heat. You can't break them down with digestion. The cells then in the body are going to, since they can't break down this misfolded protein, they're just going to kick it out of the cell because it's misfolded. They can't do anything with it. So the cell then kicks the misfolded protein out of the cell, which then goes and interacts with other misfolded prion proteins outside the cell. And then they start to form these extracellular plaques. These plaques are actually physical structures that you can detect on the brain. And once these prion plaques start to form and interact with neuronal tissue, they're going to block neuronal communication. And once you shut down the brain's ability to communicate, you're going to lose a lot of normal functions within the brain. So what are some of the diseases that are caused by these prion proteins? Well, in humans, if it is a sporadic prion disease, so this is essentially, let's say you consumed a misfolded prion protein. For the purposes of the example we're discussing here, since we're talking about mad cow, let's say you ate a hamburger that had some prion protein in it. You've now sporadically introduced prion proteins into your body, which means you're now going to have a sporadic origin of prion diseases. And this is going to be where now your normal prion proteins are going to be converted and misfolded by the bad prion proteins that have already misfolded. This starts a snowball cascade where more proteins misfold. These misfolded proteins get exported out of the cell. They start to form these extracellular plaques. And so we see some diseases that we can actually link back to the sporadic origin, such as Creutzfeldt-Jakob disease, human protease sensitivity, prionopathy, scrapie, and then bovine spongiform encephalopathies. So these are prions that are misfolding just sporadically and bringing on prion diseases. We also have a category of prion diseases that are genetic in origin. So there are certain individuals out there that have certain genetic mutations that make their prion proteins very prone to this misfolding. And once they misfold, again, it starts a snowball cascade, which is eventually going to lead to prion disease. So within the diseases that are genetically in origin, have a genetic origin, we see a form of Creutzfeldt-Jakob disease that can be inherited through your family. There's another inherited form of prion disease called fatal familial insomnia, which we'll talk about, which, as the name implies, brings on uncontrollable insomnia. There's a rarer form of prion disease called Gerstmann-Straussler-Scheiker syndrome, GSS. And these are all going to be genetic in origin. So these are not diseases that were caused by individuals consuming misfolded prion proteins. These are individuals that have genetic mutations in the gene that encodes the prion protein, which leads to the production of these misfolded prion proteins. And then lastly, there are acquired forms of the disease. These are prion proteins that have been acquired through either the consumption of infected beef. They can be introduced through hormone injections or through certain tissue grafts. We have people that have converted into prion diseases through blood transfusions. Of course, if you are a cannibal in Papua New Guinea and you happen to consume the brain of your enemy, you could come down with prion disease, which is called kuru. And then we have some other forms of prion diseases down here that can be acquired from consuming infected meat, for example. So prion diseases can either be sporadically introduced, they can either be genetic in their origin, or they can be a disease that you acquire over time. And now we'll go through some examples of each one of these types of prion diseases. So what are some basic characteristics of all the prion diseases, regardless of their type or how they're acquired? All these prion diseases are going to be confined to the central nervous system, which means they're going to have to get into the brain. They're going to have to get across the blood-brain barrier some way. They all have a very prolonged incubation period. If you get infected with prion protein, you're not going to start displaying symptoms the next day or even the next year. This could be a disease that might take years before you start to show any symptoms. So the incubation period is very prolonged. Again, the prion protein and prion disease is caused by the normal prion protein misfolding and then sharing that misfold and inducing other normally folded proteins to misfold as well. So if you have one misfolded protein, it's going to take time for that misfolded protein to convert 100,000 other normal prion proteins to be misfolded as well. So it has a very prolonged incubation period. Prion diseases all have a very slow progressive course, which is tied back to the incubation period, but they're all fatal. We have not seen to date a single survivor of prion disease. If you come down with a prion disease, it's going to be a fatal disease, and it's going to have a very slow progressive course. And then there's some anatomical structural changes that we see that are indicative of all prion diseases. All prion diseases appear to show some kind of spongiform encephalopathy, and they all seem to show some kind of vacuolation of neurons. So essentially, destruction of neuronal tissue. So regardless of the type of prion disease, whether it's acquired, genetic, or sporadic, they're all going to have these same 5 characteristics. So as again, just kind of to indicate again, we're talking about a protein that has a normal physiological function that somehow is induced to misfold, and when it misfolds, it's going to lose that normal function because again, for proteins, shape determines function. So when the prion protein misfolds and it loses its normal function, it's now going to be this abnormal protein designated by this PRPSC nomenclature, which is now going to be an infectious particle. The smallest known infectious particle out there, smaller than any virus that's known. And again, we don't have a really good way at this point to be able to break down prion proteins. I've seen at USDA where they've used renderers, which are immense titanium steel vats that are buried in the ground that are about 10 by 20, 10 feet by 20 feet, big enough to hold an entire 2,000-pound steer, where a cow that was infected with mad cow disease was put into this renderer. It's basically put under pressure and filled with acid and left for 6 months. And you come back in 6 months and look what's in the renderer and nothing's left but a black goo. If you take that black goo and sample it, you will still find abnormally folded prion proteins. It's a very, very difficult protein to break down, very resistant, and it stimulates no host immune response in the host, which might be the worst characteristics of all. So if you become infected with this protein, it's very, very difficult to resolve the infection. Right now, I would tend to say it's almost impossible to resolve the infection. Again, so we've kind of talked about the replication cycle a little bit, but we have this protein that's going to be produced normally. Somewhere in its life cycle, it gets misfolded into this abnormal configuration. Once it's misfolded, it can't be recycled or can't be reused back into the cell. So it's now going to be kicked out of the cell. These misfolded proteins are going to begin to accumulate with other misfolded proteins. And once this happens, they're going to start to form these amyloid plaques. And this is going to cause the cell, generally going to be the neuron in the brain cell, to lose viability, which is going to result in the cell death. We're going to see a change in the gray matter, that vacuolar change, which we talked about, which is characteristic of spongiform diseases, of prion diseases, which leads to this spongiform encephalopathy. We're going to see vacuolation of the neurons and the formulation of these amyloid-containing plaques and fibrils, very similar to what we see in Alzheimer's disease. And again, all these changes to the structure of the brain and the structure of the ability of the neurons to communicate with each other is going to lead to downregulation of brain function and severe complications for this individual as the disease progresses. Prion proteins reach very high concentrations in the brain. When they've looked at they've done autopsies on individuals that have been infected with prion proteins, they've been able to isolate the prion protein from liver, kidney, spleen, and some in the lungs and the gut. But primarily the highest concentrations of prion proteins are going to be seen in the brain. Of the different tissue types where prion proteins have been extracted from, when they've done pathology on those tissues, no damage can be shown to those tissues. So it looks like only the brain tissue shows any pathology within the context of prion disease. And again, this protein does not generate any immune response. Scanning of the prion protein sequence to identify the ability of the immune system to respond shows that there's no part of that molecule that will elicit an immune response. So there are no pieces of the prion protein, if you will, that have the ability to contact and reach out and stimulate an immune response. So if you have a prion disease, you're not going to get an inflammatory immune response, nor are you going to get an immune response in general generated against the protein, which is going to make clearing this disease that much more difficult. If we look within the brain itself, we can kind of assign some general areas within the brain, general structures within the brain, where we see certain types of human prion diseases localized. And again, this is not going to be 100%, but from a generalization perspective, we see in the cerebral cortex, this is where we usually see Creutzfeldt-Jakob disease, where most prion are going to accumulate in the cerebral cortex if the individual has Creutzfeldt-Jakob. If they have fatal familial insomnia, we're going to see a lot of prion disease accumulating in the thalamus. And again, if you kind of think about this a little bit, how is sleep and the circadian rhythm regulated? It's regulated from the hypothalamus and the thalamus area. So if you have prion diseases accumulating in that area, it would not be unexpected if you saw disruptions in circadian rhythms and sleep patterns, which is what we see in fatal familial insomnia. These people are unable to sleep because of the accumulation of prions within this hypothalamus area. For bovine spongiform encephalopathy, we see a lot of prion accumulation in the brainstem. And again, when we think about BSE, bovine spongiform encephalopathy, and other wasting diseases such as scrapie proteins also accumulate in the brainstem. A lot of these individuals and these animals display loss of function. They lost their ability to regulate how they walk, their mobility, their balance. They display vertigo. So it's not maybe unsurprising you would see prions accumulating in the brainstem for individuals that are infected with bovine spongiform encephalopathy. We see the same thing occurring in people that consume infected beef and come down with prion disease, that we see a lot of prion accumulation in here. And then for other forms of prion disease, specifically for Kuru, we see a lot of prion proteins and GSS occurring in the cerebellum. One of the hallmarks of Kuru disease is a loss of emotional control. And again, we know from basic neurology that a lot of that areas of the brain that control emotion are located in the cerebellum. This is just some images kind of showing you under normal conditions what the cerebral cortex would kind of look like. And over here in panel A, we see a cerebral cortex from someone that's infected with Creutzfeldt-Jakob disease, and we're already starting to see the spongiform degeneration. We're starting to see subvacuation of the neurons, and we're already starting to see kind of the breakdown in the brain tissue. These over here in panel D, as indicated by these very large dark covered patches, are actually prion proteins accumulating in a patient, in the cerebellum of a patient who has GSS. So this patient was identified with GSS. They later passed away. An autopsy and pathology study that was done on the brain tissue looked at slices of the cerebellum, and they found significant deposits of the prion protein. So you can imagine, again, this is brain tissue. You start to have big chunks of foreign protein and stuff deposited in your brain, it's going to interfere with the cells where these deposits are made. Up here in panel A, if you're having massive degradation and degeneration of the brain cells of the neurons in here, you're now going to start to have gaps between the cells, which is going to be very difficult to transmit neuronal signals for your brain to tell you to do something like pick up your arm or blink your eye or take a step to the left or to the right. So this is why prion diseases can be very, very debilitating. And this is, again, something that takes years to accumulate. Getting a prion disease does not mean that you're going to come down with this pathology the next day. This is something that's going to take years to progress. And of course, there's nothing we can really do to stop the progression itself. So let's discuss some of the clinical symptoms of prion diseases, and then we'll get into some of the treatment modalities as well as we begin to wrap up session 4. So as I mentioned a couple times, the incubation period for these diseases can be very, very long. For Creutzfeldt-Jakob disease and for Kuru, this incubation period can be as long as 30 years. However, once symptoms are positively identified, clinically at least, usually within 12 to 18 months, the patient is going to die. So again, this is a very slow progression disease, but once it progresses to the point to where symptoms are visible, there's not much time that's going to be left. The transmissible, these are the inherited forms of disease or the Kuru. The transmissible spongiform encephalopathies are very rare. They're usually sporadic, they are rapidly fatal, and they usually tend to preclude dementia in a global case. We first called these transmissible spongiform encephalopathies Creutzfeldt-Jakob disease. Everything kind of got the initial name when prion diseases were first identified, kind of initially got grouped as a Creutzfeldt-Jakob disease. And now as we've gone back and studied these pathologies in more detail, we've been able to kind of segregate them out into being either sporadic or acquired. The sporadic cases are sporadic, but they do seem to about 5 to 10% of the time occur in individuals that have a genetic predisposition to fatal familial insomnia. So while the disease is sporadic, about 5 to 10% of the cases that we see pop up in individuals who have a genetic predisposition to already having a prion disease. So let's talk briefly about Creutzfeldt-Jakob. It's something that thankfully is a disease that's very rare. Its incidence is about 1 to 3 million annually. These are numbers that are global numbers that were taken from 2012. It's characterized by very rapidly progressive dementia. As shown in the cartoon here, you're going to have brain shrinkage and deterioration that occurs very rapidly. You're going to have the spongiform pathology that's characteristic of Creutzfeldt-Jakob that's going to be visible in brain sections. Of course, you probably wouldn't see these brain sections until the patient has sadly passed away. But because we're seeing these gaps in neurons, and because of where this is occurring within the brain, remember we have the nice graphic showing us where Creutzfeldt-Jakob is going to take place up in the cortex. You're going to have disturbances in behavior and the higher cortical functions. This is going to appear in just about everybody. And these are sometimes going to be precluded by confusion, vertigo, difficulty seeing, even might have headaches, clumsiness, the ability to control your movements. And again, this is something that's going to take a long time to progress, but once the symptoms appear, you're probably looking at death within 12 to 18 months. So the acquired form of Creutzfeldt-Jakob disease, that's the new variant Creutzfeldt-Jakob disease, is something that's being reported in places, primarily in people who are younger. So remember, for classical Creutzfeldt-Jakob disease, this is something we see occurring in individuals that are between 50 to 70 years of age. For the new variant CJD, we tend to see these occurring in people that are under 40 years of age. The variant form of Creutzfeldt-Jakob does have a distinctive neuropathological appearance. Primarily is indicated by more deposits of prion protein than typical Creutzfeldt-Jakob disease. This has been one of the primary ways of distinguishing between the two diagnoses. There is a lot of concern that this variant CJD might be exposure to eating contaminated beef. So primarily, we've seen a lot of this variant CJD occurring in Britain, in Great Britain, and England. And England has had severe issues with prion protein diseases occurring within their cattle populations that they use for human consumption. Here in the United States, this is one of the reasons why you see some countries limiting the import or export of beef or animal products that come from beef. Here in America, for example, we generally prohibit the import of beef from other countries unless those countries have showed consistently that they are free from prion diseases. So for example, the United States will import beef from Japan. However, Japan does not import beef from America. And one of these reasons is because of this concern about coming down prion diseases. Cattle herds in the United States are routinely surveyed for the presence of mad cow disease, of bovine spongiform encephalopathy. Occasionally, an animal is found here and there in a herd that is positively identified as having BSE. These animals are immediately pulled and culled from the herd, and they'll usually cull another 5 or 10 animals, or maybe even up to 100, 200 animals that were associated with that original animal and check them to make sure. England has been a little bit slower to catch up to this, and so we've seen more of these these new variant CJDs occurring in England, and one of the reasons might be because of this consumption of this contaminated beef. As of 2016, if we look at the number of Creutzfeldt-Jakob disease cases globally, and we compare the sporadic to the variant, some of the differences that we see jump out, as I mentioned earlier, are the age. We see variant CJD occurring in people as young as 19 up to 39. The sporadic seems to occur more in individuals that are 50 and older. The symptoms are going to be a little bit different, as well as the course of disease. If we go and we look at the gene that encodes the prion protein in individuals that come down with sporadic or variant CJD, we see that there are codon changes at position 129 in people that have sporadic CJD, and this is another mechanism that we use to delineate between sporadic and variant CJD, is looking at the positions of codon 129. What are the amino acids? In sporadic CJD, it's predominantly valine. In variant CJD, it is 100% methionine. So this protein change is one of the things that helps us differentiate from a diagnostic perspective between sporadic and variant CJD, as well as with age and some of the other symptoms that show up. And then lastly, if we look at the PRP deposits in the brain tissues of these individuals, we see that in variant CJD, lots of plaques. Sporadic, we rarely see any plaques. And if we look at the banding pattern, and these banding patterns are based on prion protein bands that have been somewhat digested and then ran on agarose gels, and then they display a very specific banding type, and this banding pattern is then used to kind of help diagnose and differentiate between sporadic and acquired or variant CJD. And we do see differences in the PRP banding patterns. When a prion protein is basically broken down as much as it can be and ran out on a gel, there's notable differences in how that protein bands between those individuals infected with sporadic versus those infected with variant CJD. And if we look at the cumulative number of cases, thankfully, as of 2016, the number of cases, and again, we're coming up on almost 10 years old, but as of 2016, we're only looking at about 222 cases worldwide. But again, out of that 222, about 80% of them are from the UK. 174 from the UK, 3 of them are secondary, 25 from France, and then we see the other 12 or 13 that are made up from the rest of the world. But again, France gets a lot of its beef from the UK. So you're talking about 199 out of 222 cases coming from essentially 2 countries using what is, in all intents and purposes, one supply of beef. Again, we kind of discussed this already, how we see that the variant form of the disease is something that seems to occur primarily in individuals that are 40 and over. And the sporadic form of the disease, it tends to occur in individuals 40 and over. The variant or the acquired form tends to be in individuals that are between the ages of 10 to 40 years of age. This is again kind of giving you an idea for how many cases of BSE we see occurring in the UK is indicated with the hashed bars over here that are in the world versus in the UK alone, and we see a dramatic difference in the number of BSE cases in England compared to the rest of the world, which would tend to, again, suggest that there must be a high prevalence of prion disease within their beef population. So in addition to variant CJD and acquired CJD, Creutzfeldt-Jakob disease, another prion disease that we see commonly occurring in humans that come down with prion disease is fatal familial insomnia. And again, if you remember back from where we showed the brain image, these are primarily going to be prions that are localizing to the hypothalamus and the thalamic region of the brain. And fatal familial is going to result in the ability of insomnia, of untreatable insomnia, severe disruptions to the circadian rhythm. You're going to have endocrine disorders, motor disorders, and dementia. And again, because of the location of the prion protein accumulation in the brain, it seems like the hypothalamus function is the target of this disease. And unfortunately, FFI is an inheritable disease where it's an autosomal recessive inheritable disease where each child can inherit a copy of the recessive gene from each parent. So in families that are known to have fatal familial insomnia, it's very common to have genetic testing done in these families to try to be able to track and predict these many children are going to come down with FFI, or this child has the gene, this child doesn't have the gene. So this can be helpful moving forward when these children eventually become adults and go out to plan their own families. They know that, hey, I have a copy of this recessive gene. It might be a good idea to make sure that whoever I partner with to have children doesn't have a recessive copy of the gene, so we don't end up creating a child that's going to come down with fatal familial insomnia at some point in their lifespan. Sadly, this is a disease that will not seem to display symptoms until about 40 or 50 years of age. And for most people, by the age of 40 or 50, you've probably already had children and passed on the gene. So, but fatal familial insomnia is another another prion disease that can be driven through inheritance. And probably one of the most famous of all prion diseases, the prion disease that put prion diseases on the map, is Kruger. This was a disease that was initially identified in the late 1950s and 60s by anthropologists that were studying a tribe of headhunters in Papua New Guinea called the Foray people. And they noticed that there were certain individuals amongst this tribe that had these tremors. They displayed severe cerebellar ataxia. They were not able to walk. They couldn't talk. And then usually when these symptoms came on, they had usually about a year or so left to live. The disease is kind of broken down into three phases: an ambulant, a sedentary, and a terminal phase. So in the early stages of the disease, there's a lot of emotional liability, it's referred to. The individual loses the ability to regulate their emotions. Again, if we think back to the image that we showed of the brain structure and where prion diseases, prions tend to accumulate, those that accumulate in the cerebellum are oftentimes going to be attributed to Kruger disease. And because the cerebellum is one of the structures in the brain that has a lot of influence over the control of emotion, when we see dramatic swings in emotion as a consequence of prion disease, this is often attributed to Kruger. So these individuals sometimes will alternate between manic laughter and just profound sadness. This is going to be very characteristic of the first phase of the disease. The second phase of disease, the sedentary phase, is when the person begins to lose the ability to speak and the ability to move. And then the third phase, the terminal phase, is essentially going to be now this person is essentially, sadly, just marking time. No treatment is available for any of these prion diseases, really, except palliative care. So try to give the individual as much care as you can. So when anthropologists first encountered this disease among the Foray people, they tried to figure out what was causing this. And epidemiologist named Carlton Gajusek kind of solved this problem in 1960 by exploring the link between cannibalism and Kruger. What he noticed was that he spent 2.5 years with the Foray tribe. And what he noticed was that when individuals died within the tribe, either as a result of natural end-of-life disease, natural end-of-life stage, through disease or through warfare, they were engaged in warfare with other tribes. What they noticed was that the women and children were responsible for preparing the bodies for the funeral rites. The men played really no role in helping to prepare the body for funeral rites, but the women and children did. And part of that funeral rite involved the consumption of some of the viscera and the brains from the deceased individuals. So again, if we're talking about prion diseases and the accumulation of this misfolded protein that can be transmitted through the consumption of meat that's infected, if you will, with this misfolded protein, oftentimes we see these accumulations of prion proteins in the brain. So if we have the women and children that are eating parts of the brain as part of the funeral rites, then it might not be unexpected that you would see the accumulation and the introduction of these misfolded prion proteins into these women and children. And wouldn't you know, women and children were the ones that seem to be most commonly infected with Kuru disease. This practice was ended by the government in the early 1980s, but again, these people are in Papua New Guinea. The government is about 300 miles away in almost a different region in a completely different region of the country. And so the government now describes the practice as sporadic. So whether or not it's ceased or not, we don't really know, but there's been a dramatic reduction in the number of cases from the Kuru, cases of Kuru from the Foray people since this was first brought to light. And again, this is kind of the classical case to kind of distinguish what this was. So just to kind of wrap things up in general, neurotropism is a very difficult nut to crack. In many respects, the brain is going to be protected from the infection by the blood-brain barrier. And if that barrier remains intact, then we're going to see a very robust protection of the brain and the central nervous system. And we should expect to see the individual being able to resist neurotropic agents. However, if the blood-brain barrier becomes compromised, then you're going to have the ability of pathogens, microbial, parasitic, or even protein pathogens are going to be able to get across the blood-brain barrier and establish disease. Remember, again, for viral infections, these are primarily going to have to resolve themselves over time, while there are some limited treatment options available for individuals that are infected with bacterial and parasitic infections. But in every case, they're going to require extreme levels of palliative care, and the course of treatment with the antibiotics is going to be generally a long-term course. So 3 to 6 weeks for bacterial pathogens, 4 to 8 weeks for parasites that may need multiple rounds of infection. So from a healthcare burden perspective, these neurotropic agents can put a pretty severe burden on the healthcare system. Even if it's a viral infection, the individual is going to need probably 2 to 4 weeks of palliative care to be able to give themselves the time to clear the infection on their own. In the case of a bacterial or parasitic infection, they're going to need probably double the time, 4 to 8 weeks, plus constant therapy and monitoring and the palliative care. And in the case of the prion diseases, there's really nothing that you're going to be able to do other than take care of that person and make them as comfortable as they can. At some point, we would hope to have the ability to understand more of the mechanism of how this protein misfold. It's received a lot of study, but we don't really have some good explanations for why it does misfold. And we've tried, there have been several attempts to try to unfold these proteins and fold them back using various protease treatments, using specialized cells to produce to run off proteins from a mutated prion gene, to study the mutation and how the proteins put together to see if there's a point where we could intervene. But to this stage, at least, at this point at least, we don't have the ability to convert these proteins back. So the best thing we can do right now is to have a very stringent system of monitoring to protect the food supply and protect individuals from the transmissible forms of the prion diseases. For those that have inherited forms of the disease, DNA sequencing, genetic counseling, and making sure that these individuals have as much information as possible is going to be the best that we can do to try to slow down the inheritable forms of prion disease. But for the transmissible forms, it really comes down to making sure we have a tight control over the food supply and that we continue to keep monitoring this. This approach has been fairly successful in the USA. We have not really had any cases of mad cow disease pop up in the USA. And other countries around the world, such as Australia and New Zealand, that prohibit the import of beef from other countries, have also been able to keep prion diseases out of their nations. So certainly it looks like safety and monitoring is a very robust approach, and it has been a successful approach. But in order for us to really be able to treat this disease, at some point, we're going to have to try to figure out what causes the misfolding, and if there's a way we can misfold it back. And if not, is there another way that we can somehow target this protein for clearance from the body itself? Because again, over time, these misfolded prion proteins will eventually make their ways out of the cell. And once they're in the extracellular space, they're going to start to form these plaques that cannot be broken down. Those plaques are going to sit on neurons, and you're going to have a hard time getting normal function out of the neuronal tissue that's got prion disease. So this is a list of the references that we've cited, and this completes short course session 4.